A very good morning and hello to everyone. My name is Senthil Sarvanamuthu. I am the founder and CEO for ONS Bio LLC. I thank Dr. Wilson again for the opportunity to connect with you all today. And thank you all for taking the time to be here. Today, myself and two of my colleagues from ROKIT Healthcare, Dai and Joshua Kim, will be talking about the 4D bioprinting as a powerful research tool and a potential therapeutic platform in the context of some emerging megatrends in science and healthcare. So beyond the basics of bioprinting today, we will also try to cover some exemplary real-life applications and case studies of ROKIT's 4D bioprinting technologies, not only from the research labs, commercial industries, and around the world, and even in the operating room. So today's talk is part of an active outreach to researchers and clinicians across the US to educate and create awareness about the potential for bioprinting technology to transform and advance their research or be applied as a platform technology for evolving novel regenerative therapies. Now, this is done in partnership as a partnership between ROKIT Healthcare, my company ONS Bio, which is based out of Solon. So I'm right here, your neighbor. And in my capacity as the sole distribution partner for the US Midwest. And our other partner, Indicate Technologies, is based out of Santa Clara, California, as the sole distribution partner for the US West Coast. ROKIT Healthcare is a South Korean pioneer and leader in regenerative medicine. Notably, ROKIT Healthcare is the manufacturer of 4D bioprinters and organ regeneration platform technologies. INVIVO 4D and 4D6 are their popular models, which have the revolutionary potential and applications in both basic and translational research, and also applications in the development of autologous cell therapies. It's also worth mentioning that ROKIT manufactures industrial 3D printers as well, and also offers genomics and single cell RNA services, among the other things that they do. And today I'm joined by my colleagues from Seoul, South Korea, Dae Lee, and Joshua Kim, who will also be presenting jointly with me, both from ROKIT Healthcare. Dae is a senior bioprinting specialist and consultant and global business development manager. And she's a passionate advocate for bioprinting technology. And Joshua Kim is the scientific officer and president in the Bio Inc. and ECM division at ROKIT Healthcare. Chris Kamimura is the head of business development from Indicate Technologies, and together we make up a team of bioprinting and 3D printing specialists that you can always refer to for any on-demand support anytime. And just a note, if you have any questions during the presentation, please hold them till the end of the presentation when we'll have a discussion. And you can also try to save the questions by dropping it into the chat box. All right, without further ado, let's go on to the presentation itself. First, I'd like us to take a look around and see what has been happening in the news over the last 2 years. Now, there are 5 events and megatrends that I'd like to draw your attention to. And this gets us closer to the values for which the bioprinting as a technology is getting a lot of attention today. In 2018, September, we saw this featured news of a boy who was living for 14 years with a new bladder made from his own cells, seeded with 3D inkjet machines into decellularized pig bladder as a scaffold. Now, this was a work done by the renowned pioneer, tissue engineer and surgeon, Professor Anthony Atala, whom I'm sure many of you have heard about. He's now the director of Wake Forest Institute of Regenerative Medicine. Now, this work and similar work by many others highlights the promise and potential for bioprinting of human tissues to transform medicine with implications for organ transplants, cancer treatment, and many other possibilities, such as better ways to develop antibiotics or drugs. And in July 2019, the American Medical Association approved a reimbursement codes for 3D bioprinting, for 3D bioprinted surgical models and tools. While these are temporary codes that will be tracked for 5 years until full official reimbursement, what this actually helps is it makes the foundation for a clinical registry of patient 3D scan data and relevant 3D modeling, which will serve as a learning resource, tutorial, and also provide the much needed impetus for active implementation of 3D printing in biomedicine. In September 2019, United States Environmental Protection Agency, EPA, announced that it will eliminate all mammal testing by 2035, becoming the first federal agency to make such a strong statement and commitment to the transition to humane research, while emphasizing the urgency to find alternative solutions to the use of animals in research. In October 2019, ROKIT Healthcare and a diabetic foot ulcer specialty clinic in India put the bioprinter in the operating room to create a streamlined process of skin regeneration for diabetic foot ulcers. This new and alternative method for chronic wound healing promotes skin reconstruction using bioinks made from patient's own adipose tissue that are printed into size-specific patches and applied directly onto the wounds. Now, these dermal patches integrate with the skin, and through early onset healing, ROKIT's mission is to decrease the rate of amputation for patients with the diabetic foot ulcer. This is a very important occurrence, especially given that the 5-year survival rate of patients who have undergone amputations is actually lower than the 5-year survival rate of many cancer patients. Last but not the least, in the most recent months of 2020, all of us know that COVID-19 has changed everything about the way we live, and most notably, we have seen that the pandemic disrupts the traditional centralized value chains in manufacturing. For example, when the hospitals got cut off from supplies of masks and ventilators because factories were shut down, 3D printing or additive manufacturing came to the rescue in some cases to manufacture these supplies on a on-demand and on-site basis. So why am I telling all this, and what happens if we put all these things together? So all these global events and megatrends that we are observing points to the fact that 3D bioprinting can react directly to decentralized demands from life sciences research and also serve as an effective medical strategy. Clearly, there is growing value for on-demand biomanufacturing, and bioprinting is quite relevant to the real-world needs and megatrends. So it is for this irrefutable connection to the real world that we are here today to talk about the 4D bioprinting technology. And in a short while, I will clarify what's the difference between the 3D and 4D bioprinting nomenclature. For the above reasons that I have been listing, it may be worth paying attention to how this technology is maturing to impact our research and healthcare workflows today. For those of you who haven't heard about bioprinting yet, this is a quick video to show the process. Bioprinting is a perfect combination of computer science, biology, instrumentation engineering, and material science. And it brings the benefits of personalization, autologous solutions, and fine details to human health applications. So though bioprinting is getting a lot of attention in the mainstream research and possibly the clinic as well, only recently, the origins of the technology go way back to nearly 2 decades. It is not totally in its infancy. In fact, if you look at this graph here, the number of publications around this technology has been growing exponentially since 2000. And today there are thousands of publications as a testament to the benefits and applications of this technology across tissue engineering, biomaterial development, regenerative medicine, and many, many more research areas. So bioprinting is a platform technology, platform biomanufacturing technology with applications for all major funded research fields. And in this slide, you will see that there is a list of publications that are hyperlinked. There has already been a lot of evidence about the ideal choice and combination of cells, biomaterials for tissue-specific applications using bioprinting. And perhaps it's conceivable in the future that journal publications will require data from 3D human tissues in between the 2D in vitro cell culture and in vivo animal or human studies. Now, it may become an important criterion for your research to move forward. And one of the major challenges in medicine, of course, is the fact that we don't have sufficient number of tissues or organs to replace the failing and aging ones in patients. And modern tissue engineering aims to indirectly replace damaged tissue with man-made scaffolds that support cell growth. And bioprinting is at the frontier of this field because it is the only platform technology that can create these tissues and organs with applications in all these different types of disease that come with aging. Now, there has already been a lot of evidence about the ideal choice and the combination of cells and biomaterials for tissue-specific applications using bioprinting. And many of these publications are quite relevant in that context. All right, then now it's time to address some of the basic questions. What are the key components of bioprinting? What exactly is 3D bioprinting? What exactly is 4D bioprinting? So let me try to demystify that. So bioprinting is an additive manufacturing process in which cells, biopolymers, and growth factors are combined to create tissue-like structures that imitate native human tissues. Because these printed living tissues are often further incubated to have them proliferate, mature, and change over time, there is this dimension of time that makes it appropriate for one to call this as 4D, because it's not just recreation of the 3D structure. Now, we also have to take into account the maturation process, the temporal aspect. So when you combine that 3D with the time component, then it becomes a 4D bioprinting. So it is, in that sense, appropriate to call it as 4D bioprinting whenever there is a case of bioprinting that involves living entities such as cells and tissues. Now, 4D bioprinting technology consists of largely 3 components: biomaterials, the bioprinter, and the design. Let's look further into what each of these are. First, let's talk about the biomaterials. Unlike the traditional 3D printer, which uses hard plastics, metal, and synthetic materials to create non-living structures, a 4D bioprinter would use materials that are biocompatible, biodegradable, that are derived from both natural as well as synthetic sources. Some examples of naturally derived biomaterials listed here, as you can see, are collagen, gelatin, elastin, chitin. These come from the extracellular matrix of human or animal cells. Now, there are other examples like alginate, sericin, and cellulose that come from seaweed, insects, and plants, respectively. We also have the hydroxyapatite, for example, that comes from mammalian bones. We also, on the other end, we have synthetic biomaterials that are biodegradable polymers with both strength and elasticity to mimic human tissues. And some of these have been put to good use in FDA-approved medical devices. Finally, you also have the composites, which are often, they combine the natural and synthetic materials to add or enhance certain mechanical or chemical properties. Now, these biomaterials can be soft, they can be low viscosity hydrogels, or hard biodegradable plastics. And when hydrogels are combined with cells for printing, then they are called as bioinks. And bioinks are, they basically serve as the extracellular matrix, ECM. When you are trying to print cells outside the body, bioinks play a very protective role, and they protect the cells from shear stress, providing them with nutrients and hydration, and give the overall physicochemical support during the printing process. So when selecting the materials for bioinks, there are a few key criteria to take into account. In the interest of time, I'll quickly mention that things like shear thinning, which affect printability, or viscosity, viscoelasticity, which protects cells from stress and also in some level also affect printability. Then we have to take into account the hydration degree that the bioink can offer, which directly affects the nutrient diffusion, and gelatin kinetics, biocompatibility, and so on. So all of these are important for when you are experimenting and choosing a bioink for a specific application. Now let's talk a little bit about the hardware component, which is the bioprinter itself. There are various methods of bioprinting today, such as the inkjet-based, laser-assisted, and stereolithography methods that are listed here in this diagram. But the extrusion method, which is highlighted, is the commonest of all, and over 70% of the bioprinting that we see today is all extrusion based, and that's what we are going to be focusing today. So one, as I mentioned, one key component of any bioprinter is the hardware itself, and the hardware's important component of the hardware itself is the print head that should be able to print both soft as well as hard materials. There are two ways that the print heads can print. One is a scaffold laden, where you print hard matrix, and the other one is the scaffold-free way of printing, which does not have any hard matrix. Now, another key component in the chambers is the bioprinting chamber's ability to mimic a biosafety cabinet, because after all, you're dealing with living cells, and you want to keep them sterile and contamination-free. So to optimize cell viability and functionality under the stress of printing, the chamber should have features such as the UV disinfection capability, HEPA filter, and some type of bioreactor capability as well, to provide the optimal environment for the cells, such as temperature and humidity. All right. Lastly, the third component of bioprinting is the design, which is the software component. So the 3D models of the objects that you want to print is often the design component. So the 3D model files, which are popularly known as the STL files, can be obtained either by manual creation using a design software, or they can be derived from a CAD, MRI scan of the physical structure like human bodies or organs, as is the case. Now, these STL files then get processed with a slicing software into standard machine language that describes how a 3D printer, or 3D bioprinter in this case, should print the job in a given three-dimensional space. It includes information on the X, Y, Z positioning, coordinates, printing speeds, the infill patterns, etc., etc. So here below, you'll see an example of the difference between the STL object and the G-code variations of the same object as interpreted by the bioprinter or the computer. All right, so now that we understand the components of bioprinting, we can put together the bioprinting process logically. The process principally involves 5 key steps. To summarize briefly, preparation, which involves designing the experiment with a clear indication of the problem to address. It includes selection of the right biomaterials, cell types, and the 3D models that the printer will use. Then the printing process itself, which is the actual printing process. The 3D models are sliced into appropriate printing instructions. The bio ink is placed in the printer cartridge, and the deposition takes place based on the digital model. And then we've got the post-printing process or step of the workflow, where you take the printed structure and do a mechanical or chemical stimulation so as to create stable and functional structures of the biological material. And this involves things like cross-linking the printed biopolymer by either thermal or chemical or photo treatment, and so on and so forth. And lastly, and second, following that, we have the evaluation step, which is very important because it is in this step that the cultured printed tissues are assessed for a structural, morphological, and functional characterization in comparison with living tissue standards, and to see how well we are able to recapitulate the structure and function. And in terms of the application, that involves making use of the printed product for various purposes, such as disease modeling or in vitro safety and efficacy testing, and also in applications like in vivo tissue transplantation, as is the case. Now, this is an example that helps one understand this process. This is an example to show how, if you were a cardiac patch engineer, then how you would apply each of these steps. So in the interest of time, I'll skip this, and you can always revisit this slide when you have a recording. Okay, just to introduce the applications of bioprinting, my colleague Daye will give a more focused approach to this section of the talk, but I will just like to give a broad categorization of the different applications that we can, how we can group them into 3 different groups. The example that I mentioned, cardiac patch engineering, or let's say even corneal tissue engineering. These are just one of many downstream applications possible with bioprinting. So bioprinting has been used in many types of research, ranging from tissue engineering, material development, biomaterial development, regenerative medicine, veterinary medicine, toxicology, pharmaceutical sciences, so on. So for simplicity and to help our understanding, we can kind of summarize these into 3 key areas, or 3 major areas of application. One is the in vitro efficacy test model. For toxicological testing, screening of novel products, whether it be a drug or cosmetic or food, and this is especially important in the context of replacing animal testing. Then we've got the in vitro study model for understanding human tissue physiology and disease modeling. Then lastly, we have got the INVIVO model for transplantation and regeneration. So the in vitro models for toxicological testing and screening of novel products, whether drug, cosmetic, or food, is a major application for bioprinting. An ideal study to evaluate the toxicity of a chemical substance to humans would require an extremely large number of human subjects to represent the diversity of humans. But we all know that this is unrealistic and unethical. So the use of human relevant tissue models offers an alternative and a bridge between the human trials and the animal studies with standardized human tissue fabrication process and a very high throughput specific deposition of test chemicals. So in that context, we've got the bioprinted human tissues being used in many cases, and an extension of that would be uh, uh, of this application, we could also use for many other types of tissues for drug testing. Second, we've got the in vitro study models for human tissue physiology and disease modeling. Traditional science has always relied on 2D cell culture and 3D spheroids, but the decades of research have shown that human tissues are hyper complex and heterogeneous and structurally and mechanically as well as biochemically. So for example, cancer or a tumor is a great example of that great complexity, and bioprinting's ability to fabricate and visually map the tissues in 3D, enabling us to better and to kind of better understand the human body and the disease mechanisms. And that would eventually lead to accurate therapies. So being able to print tumors using the 3D bioprinting would be a very interesting opportunity for application of bioprinting for a very important cause. Third, we have got the in vivo model for transplantation and regeneration. One of the most common challenges in the medicine, of course, as I mentioned before, is not having the sufficient number of compatible tissues and organs to replace in the events of trauma or organ failures. So we can print flat structures like skin and tubular structures like blood vessels and urethras. That is relatively simple, but hollow non-tubular organs like bladders and pumping organs like heart or lungs is very difficult to replicate. For such highly complex organs to be printed, the bioprinting technology provides a very high degree of precision that surpasses human hands. And if these could be made with patients' own cells, then the challenge of immune rejection and a donor shortage with organ transplantation can also be resolved once and for all. So with these in mind, here is an example of a beating heart, beating cardiac patch that was 3D bioprinted. And as an extension of this same dream, it's possible to perfect bioprinting in future to the point where we can have nanotechnology and probably smart material incorporation that could lead to smart organs that could have extended durability that would be unimaginable for any naturally occurring tissue or organ. All right, in the context of all of these we have discussed so far, ROKIT Healthcare is emerging as a key player in bioprinting and organ regeneration space through innovative products, services, and robust in-house R&D programs aimed at creating breakthroughs in science and healthcare. So ROKIT is dedicated to providing these innovative offerings to well over 25 countries around the world with more than 250 user institutions and partners currently. As a pioneering leader, ROKIT introduced the state-of-the-art bioprinters INVIVO 4D, which was the world's first clean chamber bioprinter. with HEPA and UV lamps and hybrid printheads. And they also have many other new innovations about which my colleague Daya will be talking. So now I will hand over the presentation over to my colleague Daya, so she can talk more about the applications, which is a lot more interesting than the basics that I have covered. So over to Daya. Daya, are you there?
Yep. Can you allow me to screen share?
Yes, I'll do the stop share. You can take over.
Okay. All right. So, hey guys, this is Daye, and I will continue on from where Senthil left off to talk about the real-life applications and case studies of bioprinting in various disciplines of science and health. So, as Senthil mentioned, because bioprinting is essentially a platform technology that allows you to fabricate with cell types, biomaterials, and 3D designs of your own, it has had applications in a lot of different disciplines of life science, as you can see in the lower right-hand corner, statistics from our actual Dr. INVIVO users. But something that I would like to start with is, of course, the corneal tissue engineering, which I understand to be one of the focus interests of your group at Kolai Institute. So as you would already know best, the dominating treatment of corneal blindness today is transplantation, but the main source of cornea for transplantation is based on donations, which currently is far from enough to meet the requirement. And this severe shortage has been really the main driver for promoting the studies of effective corneal alternatives. And also this has led to ways to engineer in vitro models of corneal tissue so that we could have a better model for studying preventive measures for corneal disease. And traditional corneal replacement methods have primarily included prosthetics that are made of bioinert materials, or they have been bioartificial corneas made of bioactive materials made of decellularized ECM scaffolds or ECM deposited by cells. However, there have been a lot of limitations, and these have included some potential biological incompatibility with the autologous tissue of the patient. The donor-recipient differences could lead to refractive problems. And even if we were to be using animal-derived B-cell ECM to create the scaffolds for these corneal tissues, there is inherently difficulty to remove all the toxic agents or cellular components that are part of the processing that is required in using these D-cell technologies. And so for this reason, bioprinting has really arisen to the attention of researchers to provide a more effective method for precisely controlling the shape and realizing optical functions of real cornea. And some of the key advantages that this additive manufacturing technology brings to us is, number one, you've got a much more precise control of shape and properties because this is a computer-aided design-based technology. And especially considering that the design requirements of recapitulating the corneal refractive power is really the key to recapitulating the function of that tissue, the ability to control how you fabricate the thickness or the curvature of corneal optical tissues with bioprinting gives you this big advantage. And second, bioprinting is unique compared to other traditional methods of manufacturing because bioprinting allows you to include multi-materials in one construct. And that means you could include various naturally derived as well as synthetically derived materials in order to create the optimal high strength scaffold that is also biocompatible. And finally, because you're able to also include living cells in a bioprinting platform through the syringe dispenser-based systems, it allows you to help you to achieve full layer multi-cell corneal in vitro models. And this is helpful for creating the macrostructures or the microstructures that the corneal tissue is composed of. And as we very well know, the human cornea has this very much stratified structure, having 5 layers, including 3 cellular layers and 2 interface layers. And this stratified structure gives us sort of a hint at how we could manufacture this tissue, at least in a better way than the conventional 2D or 3D spheroid-based methods. And right now, actually, in the bioprinting world, the major, I would say, the major layer of this corneal tissue that is gaining a lot of achievements, I guess, in bioprinted tissue results is the stromal layer, because it accounts for about 90% of the whole thickness of this tissue. So that is sort of the lower hanging fruit that people are starting out with. And if you see an example here, this is an actual journal publication found on the high throughput 3D bioprinting of corneal stromal equivalents. And as you can see in the figure 1, you can see that this type of bioprinting method is based upon what's called the negative support mold method. And the idea is that right now, one of the challenges in printing corneal tissues is, of course, finding the ideal bioink that can have enough structural fidelity first. But because bioinks themselves may not be able to print it and hold their shape, hold the concave shape on their own, this research group has made use of a negative support mold like this that was made with a SLA printer using resins. And this basically includes spaces for 12 corneal tissue equivalents about the size of human cornea, about 11 millimeters in the diameter, and about 500 to 700 microns in the thicknesses. So then, once this mold was created, a bioprinter using bioinks made of sodium alginate, gelatin, and collagen were mixed with human corneal keratocytes, and they were printed into these base support scaffolds and then cross-linked, resulting in structurally fidelis and bioactive corneal stromal equivalents. And as you can see in figure 6, the publication demonstrates that when these cells were printed, despite the shear pressure and the environment they were in, they were able to hold 86 to 88% cell viability up to day 14. And they also exhibited certain functional characteristics of corneal stroma, including the fibronectin and affectin, as can be demonstrated in the figure. And so, in addition to this, what's interesting is that using the same type of methodology, a biotech company based in India and having a research office in the US, I believe in California, is seeking to create a commercialized model of the same bioengineered cornea. So if you take a look at this video, they post up this data from their in-house R&D. And what's different about this from the publication before, is that they have created their proprietary bioink that allows to be printed with living cells and can also be elastic and hard enough to be sutured and stay transparent. So this is a testament to how the bioprinting technology is already sort of stepping steps further to not only stay on the bench, but into the commercialization for health applications. And oops. And going further from there, this publication talks about another method of bioprinting that is actually called fresh bioprinting. I think some of you may have already heard about it, but the idea is that fresh bioprinting stands for free-form reversible embedding of suspended hydrogels. And basically you print very low viscosity materials like collagen, which is hard to stand on its own when it's really printed. You print these into a bath, normally a thick slurry of gelatin bath, which contains gelatin microparticles and water. And so when you print directly into this bath, the collagen that has been previously dissolved in acid becomes, it meets the neutral pH of the gelatin bath, and then it spontaneously cross-links to create free-form structures inside this gelatin bath. So you don't need to make use of the negative mold structures that you saw in the previous publication. And another interesting point that this publication makes is that you can make use of certain novel materials such as peptide amphiphile functionalized collagen. And the idea here is that when you add PA to certain segments of the collagen gel, or I guess to certain segments of the collagen gel, and then you print them in the way that you see in figure A, the result is that the parts that are functionalized with the peptide amphiphile have more resistance to curving morphology. So you basically have a result of the corneal tissue sort of retaining or attaining the curvature all on its own spontaneously in sort of in the form of 4D printing. So with the advent of these types of novel biomaterials and biomaterial composites, as well as 3D printing technical innovations, there have been more and more strategies to mimic the shape and, in the long run, functional characteristics of corneal tissues in vitro. And in the next few minutes of the presentation, I would like to go over some actual publications of Dr. INVIVO users in our own community. And even though right now it might seem like these have no direct relevance to eye research necessarily, but a lot of the logic that is demonstrated in these publications will be helpful in demonstrating how this platform technology can be used both in the terms of printing techniques as well as in terms of biomaterial selection. So very quickly, I'll go over each slide, but if you guys have any questions, please feel free to ask at the end of the presentation on the particular slides. So here is an example that sort of correlates with the slide I just went over, because this talks about 4D printing. They've developed a novel hydrogel called OHADC80H, and this is a composite of hyaluronate and glycol and ADH, which basically can reassemble and integrate all on their own at room temperature. So there's no external cross-linking mechanism needed here. And so because of this 4D shape shifting feature of this particular hydrogel, the authors demonstrate that this type of self-healing hydrogels can have the potential in providing novel drug delivery systems that basically anneal or release for example, drug nanoparticles on their own when they reach a certain point in time. And this hydrogel could also be mixed with chondrocytes, as demonstrated here, making a demonstration that this could also be used for cartilage regeneration or other tissue engineering applications. Here, the authors of Dr. INVIVO have used the bioprinting technology to create size-customized scaffolds for adipose tissue regeneration. And what they have done is develop a new biomaterial called PLCL, which is a modified version of PCL, but has a much lower compression modulus than the medical grade PCL, so it's more apt for use in soft tissue engineering. And they've combined the PLCL with adipose-derived ECM and printed them into these patches that were demonstrated to have effective regeneration of adipose tissue as well as vascularization when implanted into animal models. And going on from soft tissue engineering, there's a lot of applications using bioprinting for hard patch or beading model tissue engineering. And here is an example of creating a hard patch using cardiomyocytes that have been derived from human iPSCs. And the idea here is that they choose a type of biomaterial that is amenable to elasticity. And that prime example is alginate, which is a naturally derived biomaterial from seaweed. And alginate can be cross-linked using ions such as barium or calcium, and these are often introduced in the form of salt solutions with chlorides. And so here they demonstrate the ability to print these alginate bioinks combined with the human-induced pluripotent stem cell-derived cardiomyocytes, and demonstrate them in these linear fashion, aligned, oriented fashions that allow the living cells ultimately to be able to contract like the human heart muscle should be able to. So for some reason, this video is not playing, but yeah, this was a video to demonstrate how the this heart patch bioprinted can be sort of contracting without any external electrochemical signals. And going on from there, there's also research going on to create muscle models as well. And the idea here is that instead of using the conventional manufacturing methods of PCL posts that are sort of that only have standard sizes, you can actually manufacture these PCL posts in various sizes and in various distances between the two, and you can combine them with the human or with muscle cell lines, whether of mouse origin or human origin, and create this in vitro model where the muscle cells align themselves around the PCL posts, and they have demonstrated ability to create an in vitro model for drug testing or studies of muscle physiology. Now, aside from these soft tissue and beading tissue models for studies of these tissue physiology in vitro fashions, there are also applications of these various soft tissues that can be used in the studies of novel drug development. And here is a study that talks about the creation of 3D bioprinted vascularized tumors for novel drug testing methods. And the idea here is that the group has printed a 10 by 10 by 0.6 millimeter patch, 3D printed patch using a mix of long fibroblasts and HUVEC cells, as well embedded within a composite hydrogel of alginate, fibrinogen, and gelatin. And so with the addition of long fibroblasts and cuvex, they're able to see that this tissue leads to the vascularization or the creation of vascular networks. And upon this 3D printed vascularized tissue, they included spheroids of glioblastoma spheroids, and allowed these to incubate together with the 3D printed vascularized tissue in order to result in vascularized tumors. And they've done a lot of cell viability tests, as well as quantification of the vessel areas and qualification of the vascular networks with the various proteins like the CD31 marker for endothelial cells, as well as actin, basically to show that the bioprinting technology offers a effective way for creating not only vascularized patches, but also vascularized tumors that could be a better model for a lot of the drug testing, anti-cancer drug testing. And going on, bioprinters do not just print soft materials and living cell suspensions, but they can be utilized for hard plastics or other sort of ceramic-based materials for the creation of bone tissue mimics as well. And here is an example of printing hydroxyapatite-based bioceramic scaffold that has been reinforced with strontium. And this strontium ion basically allows pure hydroxyapatite to dissolve in the human body with greater bio compatibility. And so this research group has basically figured out the ideal concentration of strontium to be included in HA for the best cellular proliferation as well as osteogenic differentiation. And they note those results in this particular publication for in vitro bone engineering applications. There's also a growing number of research groups using bioprinting in nerve regeneration using bioengineered nerve guidance conduits. And as opposed to the existing methods of using these types of conduits made with nanoporous structures of PLCL with bulk collagen lining up the insides of the PLCL, what they did is introduce collagen in oriented and aligned fashions, using the bioprinter directly onto the PLCL nanopore structure. And then they compared the autograft in mouse model, autograft nerve tissues, as well as the bulk collagen-based nerve guidance conduit and the 3D printed nerve guidance conduit. And they showed that the myelinated axons were denser and organized comparably in the autograft as well as the 3D printed group, which were better results from the bulk collagen and NGC model that showed the efficacy of introducing 3D printing into this application. And last but not least, because of the ability for bioprinting devices to offer precise depositions of various materials into sort of high resolution images, bioprinting is also being used to create novel drug delivery devices that are in the form of QR codes. And the idea here is that you could not only create a thin oral dispersible film that is easier to swallow, but it can also be printed in a QR code that could provide information about the patient. So combining this innovative drug delivery method as well as patient information delivery method all in one, bioprinting can provide a novel solution in pharmaceutical sciences as well. And it also has applications even in microbial metabolism studies using specific depositions of bacteria inside a carbon nanofiber-based shell membrane that allows you to basically monitor the bacterial cell metabolisms and chemical conversions in this network. And so all in all, the message that we have been trying to drive home is that bioprinting is a platform technology, but it's also it can potentially become a point of care medical strategy as well. If you could imagine the bioprinter sitting right next to the patient bed with the ability to print autologous solutions on demand, it offers a whole new way of delivering personalized medicines to patients. And so at ROKIT Healthcare, we've demonstrated that by applying this to the treatment of diabetic foot ulcer patients. And actually, our company has completed clinical studies in India and Korea. And we now have teams in countries like the US, Korea, and Italy to further demonstrate this. But basically, the idea is that, as Cynthia mentioned briefly at the beginning, autologous solutions are extracted from the patient, and these are combined to create patient-customized bio ink that can then be printed into the exact size and dimensions of the ulcers in the foot. And because in the last 20 years, there really hasn't been very much advances in the treatment methods for DFUs, other than dressings or decellularized animal ECM or negative pressure wound therapies, this regenerative method offers a novel strategy. And the results we have seen are that the wounds that have stayed open from 6 months to 12 months have closed using this method. And it offers a vision of what we call a 3D scan print system. And so the idea is that right inside the clinic, if we can portably 3D scan the wounds and apply them to a software that basically creates 3D printable images right away, that could be used as a point of care strategy. And that's demonstrated here in this video, where this is a silicone model of a DFU foot that is being portably scanned. And this image is directly being translated to a software that converts this file into a STL file and then a G code, which is an instruction for the printer to print. Sorry, this video is freezing for a little bit. But yeah, it's freezing a bit, but towards the end, it basically shows that this file can then translate into the dual dispensing of PCLs, medical grade PCL scaffolds, and then autologous human bio inks, and combine them into a implantable patch. So, with all that I have said so far, all of these applications are constantly being developed and further matured through the help of a lot of the institutions that are using this system around the world. And the uses here range from academia to industrial research institute, as well as hospitals so far. And in the last 2 minutes or so, I'll quickly go over what since we are not able to live demonstrate these devices, I prepared some images and videos of what bioprinting devices look like. So here is an example of Dr. INVIVO 4D, which is our first generation bioprinting device. And it basically is notable for being the world's first clean chamber bioprinter technology that has HEPA filters and UV lamps for providing a clean bench environment. And it's a hybrid device, hybrid material device, because it's able to print both hydrogels with or without living cells in a syringe module. It can print polymeric filaments and can print polymeric pellets and powders, opening doors to a lot of flexibility with the material usage of your own laboratory research. But going on from there, we have actually, through clinical studies, learned a lot more about the requirements that would really take bioprinting technology to the next level of clinical translation. And those requirements have come from the voices of customers, the VOC of our researchers, as well as healthcare professionals. And those requirements include the ability to print in situ on curved surfaces, not just on flat modular surfaces, because the human body itself, of course, is a curved surface. Second is the requirement for a sterilization technique going beyond just disinfection. And finally, the need for a type of cell incubation device. And this way, because we're not just talking about 3D bioprinting, but 4D bioprinting, where mammalian cells are growing, proliferating, and functionalizing in this chamber of a bioprinter, we believe that it is important to have this cell incubator built inside a bioprinter. And so the result is the Dr. INVIVO 46, the second generation bioprinting device that we have released this year. And this device is a, oops. Oh, yeah. This device is a device that combines a biosafety cabinet with a cell incubator or a bioreactor with a 6-printed bioprinter. And the ability for it to use up to 6 different materials, 5 on the right and 1 on the left, allow you to create architecturally complex structures using both soft and hard materials. Due to the inclusion of the HEPA filter, as well as the bioreactor or the CO2 cell incubator, you're able to offer an optimal cell environment for the tissue, both during and after printing. And better yet, because of the ability to print high throughput in well-played formats, there are more options for us to study with biofabricated tissues in drug development and various applications that require high throughput models of various tissues. And last but not least, the diverse material capacity is preserved all the way from Dr. INVIVO 4D to Dr. INVIVO 46, where you could make use of both soft, naturally derived, and synthetic materials, as well as hard, naturally derived and synthetic materials, like the ones included here in this table. And so, with all that said, these bioprinting systems that already have applications in the clinic allow us a fast track to medical commercialization for various topics that are currently on the bench. And some of these experiences that ROKIT Healthcare as a company has had can become a useful resource for the partners that are considering the use of bioprinting technology in their own application. And so with that said, I would like to conclude the presentation by summarizing the essences of bioprinting that we've tried to drive home today. And that is the automation, the ability to create models of better physiological relevance, and the ability to fabricate things on demand. And so with all of these strengths of bioprinting, we are ultimately able to generate biomimetic structures and tissues without having to depend on certain traditional model systems that have either been too simple or too expensive or not really ideal to the system's biology that we're trying to address. And if we can imagine this bioprinting system adopted in several different regions of biomanufacturing industries, we could imagine that these types of decentralized biomanufacturing networks can have a really powerful way to impact life science research as well as healthcare delivery. And no wonder over the last 10 years, actually more like the last 6 or 7 years, these biofabrication centers of excellence have been arising all over the world. And they focus on creating complex tissues in larger and larger sizes. And ROKIT Healthcare is together with these centers of excellence to really support researchers and healthcare professionals to explore the key benefits and paradigms that bioprinting brings to our worlds of research and healthcare. And the key paradigm that we'd like to bring is the idea of endogenous tissue regeneration. So no longer relying on systems that treatment systems that rely on resolving symptoms with small molecules or monotherapies. We would like bioprinting to shift the focus towards harnessing the body's own healing potential that is both inherent in the stem cells as well as inherent in the recapitulation of three-dimensional environments that they exist in within the human body. So thank you so much for joining this presentation. And I hope that this is just the start of experiencing a revolution that's underway not only for the way we create and use model systems for answering basic biological questions, but also in the way we practice medicine. Thank you so much.
Thank you, Dae. Shiju, Sophia, Lycia, any thoughts?
Presentation from both of you. It was very elaborate for us. And I have a few questions and some doubts, like if you can actually answer them before you actually plan to give the other presentation, probably that will be, more
Yeah, we
appropriate.
can answer your questions. So just
Yes.
Yeah.
So I'm trying to understand this whole technology of bioprinting in the disease model context. So suppose if I want to develop a corneal model, let's say I am printing three different cell layers, like an epithelium, a stroma, and an endothelium.
yeah.
So these three different cell types demand a different set of growth factors or a differentiation factors for them to proliferate or differentiate and then to survive on a 3D printed cornea. And how do we ensure that?
So that's probably the my area to answer. So I was gonna actually try to explain you about the specific tissue or tissue segment specific ECMs.
Mhm.
They just briefly mentioned about humatin. Humatin is a human primary or stem cell derived whole ECM. The by meaning whole ECM, it has more than 300 components and probably also has more than like 500 signaling molecules for maintaining tissue homeostasis. So we developed the technology to extract using cell culture to extract those whole ECM without damaging them, damaging their three-dimensional folding structure. So but because just like I said, because we use cells, we can use different cell types to extract exact ECM for that specific layer. For example, for stroma layer, we can use keratinocytes. We can for epithelium, we can use corneal epithelial cells. Of course, we can use endothelial cell to make them cell specific ECMs. But that one has a very different. There's a field, specific field called metrizone. They actually devoted to analyze all the specific component of the ECM of the each organ. So if you just type up metrizone, they have maybe they just done for ECM composition analysis for cornea, all three different layers. So but the cells know how to make that perfect balanced ECM compositions for each their own microenvironment. So we respect that. We're using cell culture technology developed from ROKIT, so we can making specific ECMs and using BioInk as sacrificial BioInk layers, and we mix that ECM in to print out three individual different layers, and then and cells together, and then they actually you know we already created like very similar niche environment for them, endogenous niche environment. So they then cells start like feel like home. They start producing more similar ECMs, and as time goes by, those layers, each layer become replaced by more like a cell secreted tissue specific ECMs. So that's the concept using 3D bioprinting to create the structure layer, and then purpose of this cell specific tissue specific ECM is to providing that similar environment, including growth factors and other cytokines.
Okay,
So,
sounds
yeah.
good. Just confirm if I have understood right. Like I have a stromal keratocyte that is
Hmm.
bioprinted, and the
Hmm.
stromal keratocytes are going to convert into a fibroblast, and then they are going to secrete their own extracellular matrix, such as the collagen 1 and collagen 4 and collagen 7 and something like that. So when these extracellular matrix are secreted by the cells, so these ECM is going to replace the material that you use for printing. Is that right?
There is a one side, one benefit you get from by incorporating cells in that layer. So we already creating like off the shelf the whole ECM we call Humatin from our product lines. So we provide exactly same that ECM cell going to produce, but it is actually positive feedback loop when cells to are creating ECM, because when they meet the right environment, they secrete the same composition of ECM to fill out the more empty spaces. But like you said, when the cornea get scratches or your skin got scratched or something, they have different fibroblast type. But wrong ECM deposited because for like just life-saving purposes for making scar. That is a collagen type 1 that not supposed to be dumped a lot in the skin. Supposed to be collagen 3 and 4 or 6, but because wound situation, urgent situation, they produce wrong ECM. So we try to prevent and that situation. So by providing tissue specific ECM already mixed in the BioInk, when the cell we incorporating cells and cells see that ECM, then they actually react to that ECM cell and ECM interactions, and then they start producing similar ECM to start replacing sacrificial bioink by using MMPs.
Oh, okay. Now it makes sense. Yeah, sounds good.
Okay.
And let's say if I'm printing the cornea, like I'm
Hmm.
planning to have multiple cornea models. So let's say I'm printing some 6 different corneas.
Hmm.
So do I actually, after printing, will be using this cornea in an insert or in a well and incubate with the media with all the growth factors and something like that? Or is there any uh, plate or setup that you actually advise us to use?
So I think there's a couple components required for you to keep them alive. So one is universal media.
Hmm.
So you have different cell layers, and then including endothelial cell layer, and then maybe later on, if you want a more complicated model, you probably including the nerve endings. And in that case, you want to have a sort of very universal media so you can co-culture all those three cell types at least. So that probably you want to have,
Hmm.
and then that might have a more like supplements, but because those ECM already have some enriched with the growth factors, so they will keep them in the guide, keep them healthy, and then we'll guide them in the right direction. The cells, well, I mean, the ECM, the growth factor bounded on ECM can guide cells in the right directions. At the same time, um, depends on your the purpose of research. So if you want very drug screening, then you want high throughput, kind of setting. So then you can make a small scale cornea in the insert for 48 wells dish, or at least, or 24 well dish. But if you want to transplant later for animal model, then you got to make a little bigger. Then you might want to use insert like some specially designed inserts, and then you can actually print them, and then you can suspension culture.
Hmm.
Because when the thickness get more than 400 microns, then cells in the middle, they start experiencing necrosis, so they will die without vasculature. So you might want to keep them in under 400 micron thickness, and then you want them media from top and bottom. So in that case, you have to do suspension culture type. And but for drug screening purpose, maybe you want to print out very one or two just single or double layers. It depends on the drug type. So in that case, you're making like a small very high throughput design. So actually I have to say it's up to your purpose of application.
Hmm. Okay, yeah. And one last very general question,
Hmm.
like with your bioprinter, like how much time does it take to actually print a cornea?
Good question. So we call that filling ratios. So if you want 100% filling, like a fully filled layer, then it takes more time. But if you just want them like side by side, or if you already using some of the mesh or some other material, like a transparent film, if and then like a water penetratable, like a dialysis membrane or something, then you print out the depends on like the nozzle size, and then how much pressure you put to squeeze out more ink in certain amount of time. So if you want to have a 50% filling, and then you wanna with a wider tip, then you can make them very fast. And then actually you said point out really good point, because one of the limit of the current bioprinting is speed, because if someone wants to actually build the actual organ in the operation room, after harvesting MSC or like adipose SVF, stromal vascular fraction from aspirate liposuction, aspirate lipoaspirate, then after they actually making nanofat or the enzymatic digestion to collect the stem cell and put in the ink, and then while you print it, it takes right now um, kind of long. So the a lot of companies, including ROKIT and then other competitors, they try to shorten the time. So I think this time with that 46 by printer, I think we have reduced printing time quite fast. So for example, um, 3 centimeter circular disc shaped cornea, or like 1.5 usually, let's say 1.5 centimeter cornea shape, like a circular shape. I think if we can print out with 100% filling, even for that thin layer, I think we can finish it within like less than 10 minutes, I guess. So if you want to have three different layers, then I think you can have them like at least, I mean, no later than 15 minutes or something, because it's so thin. So I think it's not a problem.
Oh, okay. 15 minutes is like unimaginable to print an organ. Like, that's fascinating.
But there's so much more requirement after printing. So how do you like maintain, what kind of bioreactor do you want to have?
Okay.
And especially when you print out, like if you don't have a closed chamber system, the essence you print out, and if you go to just next one, the first one you print out will start drying, cells start on the surface start dying. So there's like a lot of things to consider actually.
Okay. Thank you so much. I mean,
Yeah. Mhm.
um,
Any other questions?
So would you have time, Shiju and others, for to hear what Joshua has to say about the type of questions you asked? It's more geared towards the bio material, human
Yeah,
time,
it's more like a biomaterial. Like,
which is
is
the
there
other
anything
big component
to
other
add
than
to
the
that
printer.
from apart from the explanations that you give? Because I am very much convinced with the kind of explanations he gave. I mean, it makes a lot of sense to me now. And do you like to add more to that, Joshua?
um, so
How
so
much time
I
do you
just
need, Joshua?
what?
How much time do you need?
Um, I don't think I'll need a lot of time. So like basically the I try to emphasize wrap up this thing in a minute. So like everybody rocket going to a little bit different direction from everybody else, because a lot of people in the actual medical field they try to use the cells. But at the end of the day, cell have to, in order to form a tissue or organ, cell have to secrete ECM, and they have to organize that ECM for their own purpose and the micro ultra structures. So a tissue can function as that tissue. If the muscle cell does not align, then they cannot contract, then they cannot actually exert force. So the actual three-dimensional structure is very important, and then tissue, bioprinter is the very good tool for using it. So what I'm saying is um, what we do is actually we providing right ECM for specific cell types. Otherwise, like you said, cell doesn't know how to which direction to go, especially for the stem cell iPSMSC. So I believe the biomaterial is one of the really big challenge for us. And then especially for cornea, is a very complex tissue for three different layers, and then the nerve is like innervated into one of the middle layer, and then even for corneal epithelium layer has a different cell types exist. So I think when you start building real actual translatable cornea, and then for saying for the actual very precise study, I have to emphasize you have to consider those biomaterials. um, yeah, that's I try to end. Thank you.
All right. Thank you, Josh.
Mhm.
Daye, do you have anything to add?
No, I think Josh put a good ending with an emphasis on the really the combination of the advent of additive manufacturing and the advances in ECM based biomaterials would be important for engineering any types of tissues that are to be clinically translated.
It's important to be in this kind of a cutting edge technology space to make yourself more appealing to potential
Okay.
employers
Yeah,
and
for sure, for sure.
funders. Yeah. And ROKIT also supports a lot of grant writing collaboration as needed for
Hmm.
any evolving project. So keep that in mind.
Oh, that's
um,
interesting. Hmm.
so we can work on those as well. And I'm local. I live here in Chagrin Falls, not far from where you are all. So you'll see me often.
Sure. Yeah. Thank you,
Yeah,
Senthil.
and
Thank you for this
actually,
opportunity.
speaking of grant support, it triggered into my mind when you mentioned that you're interested in corneal wound healing as as well. We have some data on the use of humantin, tissue-specific whole human ECM, having been used in vitro wound healing assays. And we've also had some studies in making use of humantin in a microneedle patch for the healing of skin as well. So if you want more information about that, we could also connect you to those materials and further discussions.
Oh, that sounds good.